So our session that is organized by the International Committee is called From Multinational Trials Towards Global Gene Therapy Products, Approval Process, Manufacturing, Distribution, Follow-up, and Ethics. We would like to announce Alexandria Petrusich from Bluebird, who will be speaking on the thalassemia clinical trials. Sorry, I forgot to pass this to you.
Great. Wonderful. So good morning, everybody. My name is Alexandria Petrusich. I'm the Senior Director of Clinical Development Operations at Bluebird Bio. Axel and Evangelia, thank you so much again for the invitation and the opportunity to speak today. On behalf of all of us at Bluebird, I'm thrilled to be able to talk to you today about optimizing global clinical trials in gene therapy, the Lentilobin experience in transfusion-dependent thalassemia. So I'm going to start my talk today by talking a little bit about thalassemia as a disease, then talk about our approach using gene therapy and lentiglobin, and then finally share with you some lessons learned and some considerations that we've picked up along the way in terms of developing global clinical trials. But first, before we get started, I just want to share with you one of my favorite quotes from Albert Einstein. Concern for man and his fate must always form the chief interest of all technical endeavors. Never forget this in the midst of your diagrams and equations. So I probably don't need to tell this to this audience, but I will anyway. Gene therapy is hard. What we're trying to do is extremely complex, and especially working in rare diseases, our sample sizes are typically very, very small. What we're trying to accomplish is really the ultimate in what we would consider personalized medicine, being able to correct for a defective or a dysfunctional gene within a patient's own cells. Now that comes with a complete host of challenges and hurdles that one needs to get over. So there's scientific hurdles, there's clinical hurdles, regulatory, manufacturing, and if you're really, really lucky, you might have some commercial hurdles at the end of the day. But one of the things that we've found along the way is really making sure that we frame these hurdles in the context of always keeping the patient's best interest in our minds with respect to finding solutions for these hurdles. And so I'm going to be talking to you a little bit today about some of those experiences, and I hope that you all find them useful and helpful. So let me get started then with just giving you a little bit of background about transfusion-dependent beta thalassemia. So people who have TDT produce little or no hemoglobin because they have little or no beta globin due to a mutation in the HBB gene. It's actually quite a diverse disease with over 200 mutations that have been identified that can lead to the disease. So for simplicity's sake, I've actually grouped these into two categories that you can see here on this slide. The first category is what we refer to as beta 0. So patients who have this genotype have no beta globin production whatsoever. The second category is non-beta 0. So these patients typically have a genotype that we call beta plus or beta E, and they have significantly reduced beta globin production. About 1.5% of the world population is actually carriers for this disease, so that translates to about 80 to 90 million people. And even with some of the premarital screening that is instituted in some regions and in some countries, we still see more than 40,000 babies born each year with thalassemia. One of the things I find very interesting is just how migration is actually starting to change the distribution of people with this disease. And in particular, looking at migration that's coming from Asia, the Middle East, and North Africa into regions of the world where we have not seen thalassemia before. So the clinical picture of untreated thalassemia is actually quite bleak. Because of the inefficiency with hemoglobin production, patients usually present with severe anemia, fatigue, weakness. Children often suffer from poor nutrition and fail to grow and thrive. Patients usually have jaundice. There's quite a bit of bone involvement in terms of deformities, osteoporosis, and brittleness. In particular, organs such as the heart, liver, and spleen are affected because they're enlarged. And then finally, patients are highly, highly prone to infections. And unfortunately, it's infections as well as the conglomerate of these symptoms with wasting syndrome that ultimately cause their death. So the gold standard for treating thalassemia is a combination of blood transfusions and chelation therapy. Transfusions have a wonderful benefit and are extremely effective. They prolong life. People who have severe disease can receive transfusion therapy and live well into adulthood. And it should immediately alleviate any symptoms of the disease, in particular the anemia and the fatigue and weakness that patients often feel. It does have some limitations, however. The effects are temporary. In general, patients need to be transfused about every 2 to 5 weeks. And of course, this can have a huge impact on quality of life. Many of these patients are trying to balance their families and work and school, and so it can really be quite challenging. Patients are also at greater risk for fever, allergic reactions, and infection. And probably the biggest limitation to transfusion therapy is iron overload. But that's where chelation comes in, to try to remove that excess iron from a patient's bloodstream. Chelation therapy is quite effective. However, when it's not effective, iron overload can cause very, very serious and potentially fatal sequelae. Now, most of the deaths that we see due to iron overload are due to cardiac toxicity. So about 70% of thalassemia patients' deaths are due to that. And again, just in summary, I think that transfusion and chelation therapy is quite effective for managing thalassemia patients, but it does have a very high burden of treatment and side effects that can lead to poor compliance. So the only curative option for thalassemia patients is an allogeneic hematopoietic stem cell. It's the only therapy that actually addresses the underlying cause of thal. It does have 2 limitations, however. The first is limitations with respect to the application of allotransplants. The comorbidities increase significantly with age. So you find quite a few patients that are on the younger side of the spectrum that are able to receive and tolerate allotransplants. The second consideration is that the mortality risk is much higher for patients who don't have a fully matched HLA donor. In general, we find that the best matches are usually siblings or a first-degree relative. I think there's some great data and information that's out there and developing with respect to haplo or matched unrelated donors right now, but I'm just not sure we're ready yet to call that the gold standard. Regardless of that, post-transplant patients must remain on immunosuppressive drugs for quite some time. And this is to try to prevent graft versus host disease, GVHD, and graft rejection, which ultimately can be fatal. So that's where gene therapy could potentially come into play as another curative option for these patients. If we have the capability to actually use a patient's own cells and correct for the defective HBB gene in their own cells, our chances of patients developing GVHD or rejecting their graft are nil to quite low. So what I have here on the screen for you today is this cartoon of our lentiglobin lentiviral vector. So this is what we're using in terms of our gene therapy program for thalassemia and running our global trials. It's a self-inactivating lentiviral vector, and it actually contains a functional copy of the beta globin gene. Within this, there's a small point mutation at T87Q, and this is very, very helpful for us in terms of quantifying and allowing for detection of the lentiglobin-produced transgene by HPLC. And so ultimately, what we want to see are levels of this transgene production that will bring patients to normal or near normal levels of hemoglobin production, and this should be sustained. So I'm not going to go into too much detail on this slide. I think probably the audience in this room is very familiar with ex vivo gene therapy, but I do want to point out just a couple of pieces here. So the first piece is that we use a combination of GCSF and plerixafor to mobilize our patient cells and then collect them via apheresis. Once the collection is is performed, we send our collections to a centralized manufacturing organization. And that usually takes about 5 or 6 weeks or so to produce the final drug product. Finally, patients are conditioned using a regimen of single-agent busulfan, and then the infusion happens. Patients are then followed up in our what we call parent studies for about 2 years, and then have an option to roll into a long-term follow-up study for an additional 13 years. So I'm going to share a little bit of information with you now just about our platform and kind of share with you some history about how we were able to go into and create these global clinical trials using Lentilobin. So things started out using the HGB 205 study. This was a single-site phase 1-2 study conducted in France with Professor Marina Cavazzana. And she treated about 4 patients in that study who had thalassemia major. All of the manufacturing and transduction that was performed during the study was performed locally at her lab at Necker Hospital. From there, we got some really great proof of concept data, and we thought about things a little bit more and tried to use that and leverage to build what we call our North Star study. This had a pivotal change for us with changing from local manufacturing into using a centralized manufacturer. So the North Star study is another phase 1-2 trial, but it's a global study. So we have treated patients in the US, in Thailand, and in Australia. 18 patients in total were treated, and 2 critical pieces of information came out of this trial that I want to share with you. So the first is the role of genotypes in being able to predict response to Lentilobin. So the data that we presented at ASH last year showed about 9 out of 10 of the non-beta 0 patients were transfusion free, whereas 2 out of 8 of the beta 0 patients were transfusion free. So that got us really thinking and confirming that we would be able to have a better chance of getting transfusion independence in these non-beta 0 patients that were producing some levels of endogenous hemoglobin. The second very important hypothesis that we confirmed in this study is the correlation between drug product VCN and transgene expression with T87Q levels. And in particular, thinking about that with the beta 0 patients who have a higher hurdle to get over to achieve transfusion independence was very important. And those 2 pieces of information really set us up nicely, I think, for our phase 3 program, the North Star 2 and the North Star 3 trials. So the HGB 207 and the HGB 212 studies are running in parallel. One study is a non-beta 0 trial and the other is a beta 0 trial. And both of these trials are taking advantage of the enhanced manufacturing process to try to increase drug product VCN. They're both ongoing and recruiting patients. And then finally, we have our long-term follow-up study, what we call LTF 303. Again, this is a 13-year trial, and patients have the option to roll into this study once they complete the parent studies. So this study is important for us, I think, with being able to gather additional safety information, including insertional oncogenesis and replication-competent lentivirus data to date, of which we've seen none. And in addition, it also provides valuable information for us about durability of response. So then the big question is, how did we do this? How were we able to take these 4 studies and really start to develop a global gene therapy platform and use that to be able to generate important data for publications, for presentations, and really start to kind of create gene therapy as a global option? So I'm going to share with you now just some ideas and some considerations and lessons learned in how to conduct and how to manage these global gene therapy studies. So first and foremost, and to go back to my quote from Einstein, putting the patients first is absolutely critical. Especially in this age of information overload. That was a good one. Information, information overload, where there's so much information at patients' fingertips. So what I have as an example on this slide are some fact sheets that we put together to help support patients as well as clinical study staff with being able to talk about gene therapy, talk about thalassemia, and give some information about the clinical trials. So we only use these materials at sites and in areas where they're permitted, and everything goes through IRB or IEC review and approval before they're given to patients. We translate these documents into local languages as needed. And again, none of this supersedes an informed consent discussion, but we really have these tools together to try to augment that discussion and get the patient as well as the site staff to think and discuss options. Here's an example of a study-specific, site-specific participant binder. So we also create these for our patients. These binders contain information that are specific to each site and has a lot of kind of general information about the local area, the local study team, information about gene therapy, and calendars. And so we really feel, especially after receiving feedback from some of our site staff members, that patients and their families live at these hospitals for quite some time. And really being able to provide them with some information and guidance about where local things are, such as pharmacies or libraries or banks, et cetera, can really make their stay a little bit more helpful and pleasurable. In addition, we also have biographies within our study binders of their study team, which we hope really puts kind of a face to a name and gives patients and their families the option to reach out to these folks in case of an emergency. We also provide study flowcharts and timelines to really explain the procedures, expectations, and the time commitments. So a lot of our thalassemic patients lead very active lives. They have school, they have work, they have family commitments. So really undergoing a therapy like this, it's very important for them to understand what their timeline looks like and when they're going to be out of commission for a while. We've also put together a study website. Again, with the technological age here, we really wanted to take this opportunity to reinforce education about gene therapy and also include an option for frequently asked questions. So this snippet here is just one of our frequently asked questions that we included in our website about fertility preservation. So obviously with single agent busulfan and full myeloablative conditioning, fertility is something that patients need to consider when deciding if they're going to participate in one of our studies. We do cover the costs of fertility preservation where applicable and where we're allowed to in region. And so that's just something we feel is important to reinforce to patients. So the next lessons learned that I want to share with you has to do with regulatory collaboration. So we've been very, very fortunate, I think, to have the opportunity to work with regulators in a very transparent and a very collaborative manner. Both the FDA and the EMA we see as partners in this development project. And in particular, I think with gene therapy and kind of forging a new regulatory path for approval, this is very, very important. One of the things that's really helped us, I think, gain ground here is being able to take advantage of the various designations that are offered by each competent authority. So as an example, with FDA, utilizing our fast track and our breakthrough therapy just allows more opportunity for engagement with regulators and more discussion. Likewise, with EMA and the Adaptive Pathways Pilot, PRIME designation, things like that, please strongly consider trying to obtain these designations when you're working with these agencies. Now, I want to call special attention to some of the GMO submissions in both the US and in the EU. So in the US, the GMO submissions are really covered mostly by the Recombinant DNA Advisory Committee, or RAC, in the US. They have oversight over all gene therapy trials. However, they utilize sites' individual biosafety committees, IBCs. And what we have found, and I want to share with you, is that this can take quite some time. So you need to plan your timelines out accordingly. If you are fortunate enough to use a central IBC for this review, it can take less than a month, which is great. However, if you are using a local IBC, what we found is it can take anywhere between 1 to 4 months, which can greatly have an impact on your timelines. Likewise, in the EU, most countries have national or local GMO requirements per EU directives, and each country has something that's a little bit different. However, we found that the GMO submissions in the EU are not really customized to gene therapy. They're actually more focused on environmental directives and, thinking about GMOs in the terms of like agriculture or something like that. So that's a little bit of a challenge at times to try to navigate those waters, and I just want to share that experience with you. And so finally, with the collaboration that we've been able to have with our regulators, we put together a registration strategy, and I can't stress this enough in terms of being able to try to build an end game for yourself. So I'm confident that our adaptive pathways and our prime designations greatly helped us forge a relationship with the EMA that allowed us to pursue conditional approval in non-beta zero genotypes, and likewise with the US with our breakthrough and orphan drug designation, really coming up with a solid plan of how we are going to do this and how we're going to get Lentilobin to patients. So finally, I want to share with you some thoughts about centralized manufacturing. So as I mentioned earlier, we had a pivot point between the HGB 205 and the HGB 204 North Star study, where we changed from a local manufacturing model to a centralized manufacturing model. We feel like this created more of a global manufacturing footprint for us. It allowed us to have more control over our drug product manufacture and really remove the procedural variability that inevitably will happen at individual clinical trial sites. It allows us to have better oversight as a trial sponsor, and also provides almost like a test drive, a test run of our commercial infrastructure development. So again, thinking about that long-term end game and putting something together for it. It's not without considerations, though. You certainly need to make sure that there is a very robust supply chain and material transport logistic scheme that is available. In particular, trying to import or export apheresis products or drug products across country borders can be quite time-consuming and quite challenging with respect to the import and the export permit process. However, it can be done. It's also very important to understand your product's stability and try to co-locate your manufacturing organizations in regions near sites and near patients. That's critical. Communication and scheduling pathways also need to be crystal clear. They also need to have flexibility. So you're never going to know if a patient undergoing apheresis is going to have a poor collection, maybe they're a poor mobilizer. That communication between Bluebird, the clinical trial site, and the manufacturing facility needs to be locked down in order to communicate any last-minute changes. Finally, traceability and local regulations, including regulations such as FACT and JC, may also dictate some of the procedures. And so you really need to think long and hard about considering those regulations and how they can fit into your process. So then just to conclude, global gene therapy trials can be done successfully using a centralized manufacturing organization. Special consideration should really be paid with how we support patients and families during their gene therapy participation. Also clinical trial sites and making sure our manufacturing and supply chain is robust. The support from regulatory agencies is also invaluable. And please think long term. Please try to plan your infrastructure ahead of time. Think about it in terms of how you want to try to set something up for a commercial plan. And again, I can't stress enough, never lose sight of your biggest priority, your patients and their families. So I just want to acknowledge our nest here. So these are our clinical trial sites, as well as some of my colleagues at Bluebird Bio. And also, I want to acknowledge and thank all of the brave patients and their families who have participated in our clinical trials. Thank you so much.
We thank Dr. Petrusich, so the presentation is open for questions. Please address them on the microphone. Yes.
Great. Richard Lawnau, dentist, Alexandria. That was a terrific talk. And
thanks, Dick.
for some of us who work on really orphan diseases, it's chilling and inspiring to see the numbers of people affected by hemoglobinopathies that can be treated with this approach. So here's my question. I hope it's not a stupid one. And that's the difference between the effect in the beta plus and the beta zeros. Mm-hmm. Is it that in the beta plus there's enough residual hemoglobin to help in the treatment, or is it an immune response difference?
That's a great question. It's a great question. So in the non-beta zero genotypes, the beta plus or the ethal, there are higher levels of endogenous hemoglobin production. So then that hurdle to be able to get to transfusion independence is a little bit lower than that of what we see in beta zero patients who have little to no endogenous hemoglobin production.
So are you using a higher dose in North Star 3 than in North Star 2 to try to overcome that?
And so the vector copy number, so what we've tried to implement between our phase 1-2 studies and our phase 3 trials is an enhanced manufacturing process to try to increase our drug product vector copy number, which we've shown has an increase in the production of T87Q levels, the transgene hemoglobin. So really trying to get that level to something that would make those beta zero patients who produce no endogenous hemoglobin transfusion independent.
Great.
Thanks.
Okay. Thank you for sharing this nice overview on your product. I was wondering regarding the long-term follow-up. Mm-hmm. So you mentioned that this sort of voluntary participation to the long-term follow-up in these studies. What about further observation time? I mean, do you have discussion with them or FDA on how to monitor the patients when they will become adult and so on?
Yeah, absolutely. So it's a great question, Dr. Ayuti. Thank you. So the long-term follow-up study is actually conducted under our IND and per our clinical study protocol. So we have a schedule of events in that protocol that dictates when they're going to come in for additional safety monitoring, efficacy monitoring, and in particular, I think with ISA and RCL monitoring, we have built that into the protocol to comply with regulations as well. So in total, between the parent study and the long-term follow-up study, patients are under surveillance for about 15 years.
Okay. And do you envisage to have a registry for following these patients in the future once the product will be approved?
Yeah, so we're thinking about it. So that's definitely part of the plan.
Hi. I'd like to make a comment about the first question. As a matter of fact, in 2010, we published a paper showing that there is a clear relationship between vector copy number and the outcome of gene transfer in patient beta 0 versus beta plus, beta 0 plus, using more than 30 patients. So this was predicted, actually. So my follow-up question is the following. You say there is an improved manufacturing system now to increase the vector copy number. Mm-hmm. Can you share any information about that?
Unfortunately, I can't right now. That's proprietary information with Bluebird.
Oh, okay. I mean, I would say just that this would be very helpful for other clinical trials because there are people dealing with vector copy number, lentiviral vectors. So I think it's an important ethical information you should disclose eventually.
Thank you.
And my second point is, so you're kind of saying that there is room for improving the vector because in beta 0, you need to get to higher vector copy number in order to correct the phenotype. Mm-hmm. Is that correct?
So improvement in the vector or improvement
vector
in the
ability to express the
transgene?
beta globin, because obviously you cannot cure the beta 0 patient unless you get to much higher copies.
Which is exactly what we're trying to do.
Right.
Yes.
Okay, thank you.
Hi, I'm Dr. Savita Rangarajan. I'm a consultant hematologist in the UK. Two questions. Mm-hmm. One, did you have any immune responses to any of these in the gene therapy patients? And secondly, were the hemoglobin levels that you achieved in beta 0 or in beta plus, were they any different?
Between beta 0 and beta plus, well, let me start out with your first question. So there were no immune responses that I'm aware of following administration of lentiglobin. The second, in terms of the transgene, are you asking about the transgene
The levels
expression?
of hemoglobin achieved in beta 0, beta plus.
The total levels?
Yes.
Yes, so there were. We did see higher levels in the non-beta 0 patients because there was less of a hurdle for them to get over in terms of their endogenous hemoglobin that they already had. And this was reported out at ASH last year.
Thank you.
I have a quick question. Do you anticipate that by reaching higher vector copy numbers and you hopefully get transfusion independence in beta 0 patients, this would allow for reducing the intensity of the conditioning?
It's a great question. I certainly hope so. And I think it's something that we need to strongly consider and think about in terms of our myeloablative regimen. And it's something I think we're actively considering in terms of alternative conditioning techniques. But we really want to have our proof of concept with busulfan and full myeloablation first before we think about some of these other options.
Thank you.
Great. Thank you.